There's an important study that came out earlier this year in the journal Neuron, and I want to bring this psychedelic drug to the forefront to talk about. Psilocybin is a really interesting psychedelic drug that's showed promise in the treatment for depression. And this drug is actually, because of its potential as a neuroplasticity modulator, meaning promoting plasticity, has gained some attention. There's some really important considerations to think about. So we know that in depression, because I should say that the context for this study was largely in the setting of depression. One of the changes that happens in the brains of depressed animals, and we think humans, is that the number of neural connections, the number of synapses becomes smaller, becomes less. So the increases in the density and strength of the neuronal connections may have underlying impacts on behavior. And this drug is potentially a way of rapidly improving those connections. So this psychedelic drug has, again, gained some notoriety because we believe it's a way of, at least in the brain of a mouse, promoting new neural connections in the frontal cortex of mice. It may have a role in depression research. We know that within 24 hours of using this drug, researchers detected or found an increase in the number of dendritic spines. That's these little things here. So this is a dendrite, and you can see these little bulbs, these little puncta. These are synaptic bulbs at the end. These are the actual structures that make the connections from one to the other. These are the connection points that transmit a signal. And so the researchers were thrilled to see that they saw an increase in the number of these dendritic spines, both with respect to their size and their density, making them think that maybe these changes can be long lasting. So this drug is a potential way of modulating or changing the nervous system in a way that's drivable or inducible. There are really important interactions between brain tumor cells and neurons. And we know that, again, supercharging a neuron, making a neuron fire, can actually make some brain tumor cells grow faster. So though this therapy may be potentially helpful for making new connections, which showed in this mouse model of behavioral change, those new connections could have the exact wrong effect in the setting of brain tumors or brain cancer. So increasing the number and density of these connections, the effect of that is really unknown in the setting of, for example, glioma and certain types of brain metastasis. So there definitely needs to be preclinical and clinical experiments, meaning experiments in both mice and humans, to flesh that out and to understand what might the effects be before that could and should ever be tried in humans. I would definitely not recommend this treatment at this point, but it is an important consideration and something to think about as a way to drive the nervous system to make more connections that could help cognition, again, with the caveat that it could have bad effects on tumor growth if we don't understand the process well.